So basal ganglia, we just talked about last time, that plays more of a role in helping you to initiate movement, helps you to switch from one pre-program motor program to another, helps to regulate some overall muscle force and muscle tone. Synergies, it helps tap into the synergy movements that we commonly use. That's basal ganglia. Now we're going to do cerebellum, so I just don't want you to get those confused. So cerebellar is going to look more at, adjusting movements on the fly, making error corrections, comparing intended movement with actual movement. What does the cerebellum do? It does a lot. Just look at the anatomy of it. All the little folia, those little folia, it's more richly folded and then your cerebrum, and it's just really lots of stuff compact, and there's processing massive amounts of sensory information. All of the information from your Golgi tendon organs, joint receptors, muscle spindles, all that copies of that all go to the cerebellum, okay? So it does compare what you're actually doing with what you intended to do. So, when you make a movement, you typically have an idea of what you hope that movement to be from past experience and so forth, and what you expect the environmental conditions to do. But then as you do it, there'll be slight errors. Your cerebellum quickly tries to make adjustments on the fly. So there's always noise in our system. Every movement we make has a little bit of error, but most of the time it's good enough to get the job done. And especially then when you get into things like very high-level sports activities, there's a reason why you practice, and you practice for 10,000 hours. You're training your cerebellum to get really good at correcting every minutest error in a stereotyped movement that you're trying to occur. It takes that long to get it, so it's especially, sports that require very specific, precise movements and so forth, okay? It's essential for good coordination. It's especially important for equilibrium during rapid motions. So things where you're making rapid changes in direction, rapid motions where you don't have time for conscious awareness of stuff to occur to make corrections to your coordination, to your equilibrium. So it's really important at the subconscious level for making corrections in equilibrium during rapid motions. It's also really most important during movements where it helps to dampen rapid pendular movements. So anytime you're making rapid movements, especially multiple ones in a row where you're turning muscles on, turning them off quickly, it helps to smooth, initially helps to boost the muscle activity, but then really quickly afterwards it helps to down-regulate the muscle and turn it off in a very pre-programmed, smooth, dampened way, so it looks smooth when you turn a muscle on or off, when you move from one direction to the other, and you're doing things quickly. That's where cerebellum is really important when you're doing this kind of stuff. Quick movements, pendular movements that require turning the muscle on and off and braking it. So you're turning it on, then you have to turn the brakes on, and those kind of things, going back and forth between agonist and antagonistic. And it's essential, I should say essential for motor learning, not just important, it's essential for motor learning. Patients that have cerebellar damage, it is really hard to get them, if they have big cerebellar damage, it's really hard to actually get them to learn new movements. And a lot of times you're really having to rely on ways of training them that are compensation-based. I may not be able, if somebody has massive cerebellar damage, they're really ataxic, I may not be able to improve their balance or the ability to perform a certain functional activity. And then I may have to actually give them some AFOs. Maybe I have to actually block their ankles. So I give them knee braces or AFOs to minimize movement because they can't control the movement themselves. Or I give them a walker or a cane, those kind of things. I may not be able to improve them. However, most people don't have complete damage of the cerebellum, and there's still pathways that are still working. So most people, you can get some improvement, but eventually you might have to do some compensation. So the anatomy of the cerebellum, let's talk about that. So kind of like your cerebrum, the outermost surface is gray matter. Right? Our cell bodies and our cerebrum are on the outside, and so are they in our cerebellum, okay? It has 3 layers. It has a molecular, a Purkinje, and a granular layer. The outer and inner layers, okay, so the molecular and granular layers, contain interneurons, lots and lots of interneurons, okay? Obviously, the granule layer contains a lot of granule cells, and then there's also these things called stellate and basket cells. Just recognize, if you hear those names, those are just cells that are those are interneuron cells within the cerebellum, okay? The granule cells are interesting, is that they project up, they're on the inner layer, they project up some axons, and those axons run parallel to the folia in the surface of the cerebellum. We'll mention those a little bit here later. Golgi, stellate, and basket cells are all inhibitory in nature, okay? The reason that you have so many inhibitory cells, these interneurons in the cerebellum, is that it basically creates this thing called lateral inhibition. Basically, what it helps to do is when you have discrete signals from your muscle spindles and Golgi tendon organs, and they're coming in from a very discrete region of a joint or a muscle, and they go up to your cerebellum, that you want it to register in a very specific location in your cerebellum, and then you're going to send a signal from your cerebellum back out to the motor system to make any corrections, and you want those corrections to go back to that certain location. So you want all of your information in these very discrete little packets throughout your cerebellum. You don't want a lot of crosstalk between these little packets of information. So all these inhibitory interneurons that are laid out there, when information comes in, they can't spread across the cerebellum going on. So and interact with whatever little Purkinje cells that are going to be your output cells from the cerebellum. It's only going to go to the ones they need to go to and not go to other ones. So that just helps to divide and package, called lateral inhibition. Okay. The middle layer, the important, they're all important, but the most important layer is the Purkinje cell layer. This is the output cells of the cerebellum. So information comes into the cerebellum, it gets processed, but the only cell that projects out of the cortex, from the cortex into the deep cerebellar nuclei, is the Purkinje cell, okay? Then we need to talk about also, how does information get into the cerebellum to the cortex to be processed? And that's through afferents. So there's a couple types of afferent pathways of getting into the cerebellum that bring information to the cerebellum. Two major types of fibers, they call these. They call them mossy fibers or climbing fibers. Okay? So the mossy fibers are all the fibers that are bringing in information from your, basically, spinocerebellar tracts, right? All those spinocerebellar tracts we talked about, anterior, posterior, kineo, spinocerebellar tracts, rostrospinocerebellar tract, all those ones. As a group, they all are bringing information from the muscle spindles, GTOs, joint receptors, all come into the spinal cord as mossy fibers. There's information from your vestibular system that also goes in. Your vestibular system is a very important sensory organ, just like your joint receptors, but this tells us about head position in space. Those also go into the cerebellum via these mossy fibers. And then we also have the pontine nuclei, which I will, later on, I'll kind of draw out with the pontine nuclei. The pontine nuclei is in the front of the pons. They are bringing in a copy of your motor tracks or your motor message that's going down, and that's how it's delivered into your cerebellum. But I'll draw this out so you see this, okay? The other is the climbing fibers, okay? And the climbing fibers come from a spot in the medulla, in the two little bumps. Remember the olives? Those are and inside the olives, those little bumps on the outside, when you take a cross section, you're looking down in it through a horizontal section. It's called the inferior olivary nucleus. Okay, and we'll see a cross section. I don't know if we'll see it in this, but at a later point, it's very, it's kind of like a little worm in there, and it's very, kind of got lots of convolutions to it and so forth. It's a very richly innervated structure. So the inferior olivary nucleus is almost like a little mini cerebellum outside of the cerebellum, but in your medulla, and the inferior olivary nucleus also receives copies of all of the sensory information coming up from all those Golgi tendon organs, muscle spindles. It gets a copy too, and it also gets a copy of all the motor pathway information afferent information coming down. And it acts to compare those two. So it's like a little mini cerebellum making comparisons between sensory and motor output, and then it also delivers that up into the cerebellum. Okay, so when we'll talk about the role it plays in adjusting more long-term sensitivity, excitatory and inhibitory influence over the Purkinje cells. Yeah. I'll show you. Yeah, so we'll talk about that. So yeah, I'll show you how it gets from the cerebellum back up to the cortex. It uses the thalamus. So it's processed in the cerebellum, and then the cerebellum sends a signal up to the thalamus. Then that goes back up to the cortex to influence the next motor signal coming down. They are. They go up to the cortex to influence the primary motor strip. But you're not. But it's not influencing the sensory, so you're still not consciously aware of it. But it has to influence the next motor signal coming down. So it also influences some brainstem pathways too. Some of it doesn't have to go up to the cortex. It can influence some brainstem, especially the vestibulospinal tracts and the reticulospinal tracts for more automatic postural gross movement associated with head and vestibular input. So. This is just showing you a little schematic of the 3 layers of the cortex. So that middle layer, here's the, oops, why is it not giving me the option of ink? Huh. Giving me a laser pointer to do stuff with. I'm going to really need the ink here in a second. Pen, why is it not giving me that option? Let me try something here. See if it works in this one. Nope. Escape. Let's go back into it. Wow, this is not good. Should have got a Mac, right? Saved. Oh, protected view. How do I? Oh, I see. Look at you guys. So do I need to how do I get out of protected view? What? Hey, yay, sweet. Now let's see if, okay, looking like it's doing something. Nice. Thank you. Oh, yeah. Oh, I don't want a highlighter. I want the pen. There we go. All right, Purkinje cell. There's a cell body for Purkinje cell. So this is the inner layer, is the Purkinje cell layer. Here's some Golgi cells. Here's that granule cell right here. And that granule cell, as I mentioned, here's a mossy fiber coming up, bringing in afferent information into the cortex, communicating with the granule cell. Granule cell is here in the granule layer, that's innermost layer, but it projects an axon up into the molecular layer. And then it has these, it splits, its axon splits and goes in different directions. It goes about 1 to 2 millimeters in each direction. But what that does, it allows it to communicate with several to several hundred Purkinje cells, okay? And they're all kind of lined up in this little packet, so it's communicating with a select number of Purkinje cells, delivering that information coming from a select area of your body through that afferent input from the mossy fibers. And remember, just the mossy fibers, I think of mossy as most. It brings in most of the information from almost all regions of your body and your vestibular system through the spinocerebellar tracts or from the vestibular system coming right into your brainstem and then into your cerebellum. Okay. All right. Without trying to give you too much detail, but giving you enough to understand some of the role of the cerebellum, your book does not go into this. But I'm just going to mention a couple little quick things here. So this is a single functional unit of the cerebellum. We have a Purkinje cell here, okay? A Purkinje cell always delivers an inhibitory impulse, okay? A Purkinje cell at base is always firing. So you have a certain level of firing rate that it's a basal firing rate that your Purkinje cell is firing at. And you can either turn it down and make it slower in its firing rate so it's less inhibitory and therefore more facilitatory, okay? So you can either turn it down, that base firing rate, or you can turn it up and make it more inhibitory, okay? So that's how we adjust the output of the cortex or of the cerebellum. Purkinje cells communicate with these deep cerebellar nuclei. We're going to get into the deep cerebellar nuclei, which are the dentate, the vestigial nucleus, the globus and umbelliform nuclei. So these are nuclei that are down inside of the cerebellum, almost kind of like basal ganglia nuclei in the middle of the cerebrum. These guys are down deep inside the cerebellum. Okay. So what happens is, well, I'll show you a picture here in a second, draw it out. But here is mossy fibers coming up into, these are the afferent fibers, bringing in all that information from your sensory receptors, proprioceptors, and those kind of things via the mossy fibers, okay? We also have, this is the inferior olivary nucleus, so those are the climbing fibers. The climbing fibers are also bringing in a copy of all that information via the inferior olivary nucleus, because the inferior olivary nucleus is like the little mini cerebellum down in the brainstem, in the medulla, that also received a copy of all the spinocerebellar tract pathways, okay? The connections, they have these collaterals that come off. These connections are excitatory to the deep cerebellar nuclei. They go up to the granule cells, at least the mossy fibers do. The mossy fibers go to granule cells, they're excitatory, and then they go to parallel fibers, which are also excitatory. The climbing fibers basically climb up and almost wrap around, very intimately wrapped around and connected to the Purkinje cells, okay? So, let's talk about first of all, like a rapid pendular movement. When you make a rapid movement that requires you to turn muscles on quickly, but then turn them off in a controlled way. And it's happening so fast, it's relatively unconscious. This is what kind of happens. You have what's called the deep excitatory loop, and then you have what's called the cortical inhibitory loop. So first of all, when I start to make a really rapid, fast movement, I turn my muscles on. So I turn my bicep on quite strongly, okay? But very quickly, I'm going to have to turn it back off because it's only going to be on briefly. So you get this brief amplitude boost. And so as you start to turn the muscle on, initially what happens is a copy of that motor signal is delivered immediately to the deep cerebellar nuclei via the inferior olivary nucleus and from the mossy fibers. And then that excites the deep cerebellar nuclei and sends an excitatory signal. The deep cerebellar nuclei, in this case, we're going to say it's from the dentate, which goes to your limbs. It sends a excitatory signal back up to the thalamus to actually increase the motor output. It says, hey, I'm turning on this muscle quickly and powerfully. I'm going to go ahead and send a little booster signal to it to keep it firing. But it also knows very quickly you're going to have to turn it off because you can only move your muscle and your joints through so much range before you got to turn them back off again. And so that signal, so let me just erase some of this stuff. So you initially get this little excitatory booster signal, which is called the deep excitatory loop, because of those initial excitatory connections of both the mossy fibers and the inferior olivary nucleus, okay, which have received a copy of your motor signal that you were sending down to that muscle. It goes back in there and says, boost the signal a little bit. Let's boost it. He's wanting to do something quickly and powerfully with that muscle. Let's give him a little booster signal. But then there's a little delay because now the signal continues up to the granule cells and also continues up to the dendrites here of the Purkinje cell. And now you get this excitation or increase in the inhibitory output of the Purkinje cell. A fraction of a milliseconds or few, milliseconds later, that turns the muscle off, okay? And it does it in a controlled way so that you turn the muscle on, but then you turn it off in a controlled way. And that's called the cortical inhibitory loop. So you get this brief excitation initially where it helps the muscle do its job, but then a few milliseconds later you get a dampening and a turning off of the muscle, okay? So remember I said that Purkinje cell is always going boop, 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 maintaining a certain level of resting activity in the cerebellum. You can then, let's say you start to do an activity and you do it repetitively, and your muscle starts to learn, it says, hey, next time that happens, that wasn't quite right. I want to make sure that I turn that muscle on a little harder and turn it off a little more powerfully the next time. Well, that is done by adjusting the sensitivity or the overall activity of the Purkinje cell. When the inferior olivary nucleus sends its signal up here, it actually lasts much longer. It's called a complex spike. And over time, the inferior olivary nucleus changes the resting set point of how much negative inhibitory impulses are coming out of that Purkinje cell. So then when the mossy fibers bring their information to add on to it, the amount of output from the Purkinje cell is already, you set the Purkinje cell at a new level so that the next signals coming in will be influenced by that new set point. So you're adjusting the sensitivity of the Purkinje cells. So the climbing fibers coming up help to really adjust the sensitivity of the Purkinje cell, especially with like repetition and learning, okay? And that's because your inferior olivary nucleus, it's getting a motor signal and a sensory signal coming up, and it's comparing those two to make sure if it notices that there's a mismatch, it goes up and sends a signal up to the Purkinje cell to adjust the Purkinje cell up or down, so that when the mossy fibers bring their signal up, then the next time you have to adjust something, it's probably going to be closer to what you would hope your movement to be. So it's just a matter, think about, now let's just kind of wrap it all into just one simple kind of statement here. Climbing fibers help to adjust the sensitivity. So climbing fibers, the inferior olivary nucleus, help to adjust the sensitivity, resting kind of sensitivity of the Purkinje cell, which is always inhibitory. The mossy fibers bring in information from the body, okay? And they then will influence the Purkinje cells at any given moment. And we have that deep excitatory loop and that cortical inhibitory loop. Again, cerebellum is most important for rapid, pendular, dynamic, quick movements. When you do something very slowly, your cerebellum isn't that active because there's plenty of time for you to use vision and even conscious sensation to make adjustments to movements. But this is kind of a little bit how the cerebellum works with this circuitry. I'm going to draw it out in a more kind of large format here in a second. So, any questions on that? I'll go to you then.
For rapid movements, we only get certain amount of time. So say instead of doing like a quick jump, doing like a 40-yard dash, it's still rapid relatively because the cerebellum still clarifies.
Yes.
Running a marathon, even though you're still moving fast.
It does. It does to an extent. However, the more you learn or do a movement, the less active the cerebellum. Once it's become a very stereotype movement, cerebellum doesn't have to do as much. And there's certain types of movements like running or walking where a lot of the coordination happens in the spinal cord level. It only happens, the cerebellum gets involved when you're running a marathon and then somebody bumps you offline. Or you like step in a hole and you have to make a corrective action to keep your balance and those kind of things. But if you're just running very stereotypically straight ahead and you've done it before lots of times, then your cerebellum's pretty quiet when that's happening. So it's only when there's a change. But when you're initially learning a new movement and learning a new pattern of movements, cerebellum's trying to make all these adjustments to set those Purkinje cells to the right level of activity and learn as you go from the errors that's going on to now set those Purkinje cells to the right level of sensitivity so that you don't overshoot your target and you don't undershoot, and you turn the muscles on and dampen them in a smooth way. It does quiet down, yeah. When you master a task, you don't have to use your cerebellum nearly as much. But you never quite master a task. You're always, there's always a little error. Even Tiger Woods has to work on his swing all the time because little errors come up. Because what happens is you get older, or you sprain an ankle, or you get a stiffness in a joint. Now you've got to change the way you did that activity and your cerebellum plays a role in that. Anytime, or the conditions change. It's the wind comes up, or you're on a different surface. So the cerebellum's always going to help a little bit. I'm going to go to Dennis next. So yeah, so they work together. Cerebellum is making adjustments on the fly based on conditions. When you learn a task, then you get a store for the synergies, and the basic movements are in the supplementary motor area and premotor areas, okay? But the cerebellum is really important initially when you're learning the task. You're storing it both here and getting it refined in your cerebellum. And then your cerebellum is now important for that task if the environmental conditions change for that task, yeah. So, good.
So you said Purkinje cells are always inhibitory.
Yes.
But adjusting the sensitivity will make a muscle turn off quicker.
What's that?
Like adjusting the sensitivity will make a muscle turn off quicker or inhibit.
It would be inhibitory back to the motor cortex. If you made this Purkinje cell more inhibitory, it would inhibit the motor thalamus. You make it less inhibitory, it's got less inhibition over the motor thalamus and over the motor system, so you get more motor output. It's always inhibitory. It's just either more inhibitory or less inhibitory. That's how it adjusts the sensitivity. And the amount of inhibition that it provides with each action, potential, or pulse is adjusted by the climbing fibers, the long-term sensitivity.
So you're saying like when you lift heavy objects, that sensitivity kind of changes?
Yes. Mm-hmm. Yes.
So is it desensitized, so it's less inhibitory, or how does that work?
There's a combination of things that happen. So I mean, if you're lifting heavy objects all the time, you can turn up the expectation to give you a little more powerful input back to it. But there's also things that happen at the neuromuscular junction in the periphery where you end up delivering more neurotransmitter, or you recruit just the right, muscle fibers that are optimum for the job. So it's a combination of those two. So, what's that? So if you get feedback every time you lift like a really heavy object from the muscle spindles in your bicep, you get the muscle spindles going up these climbing fibers, because your inferior alveolar nucleus also receives copies of all that proprioceptive information. It comes up and says up to the Purkinje cell, and it actually would downregulate the Purkinje cell and make it less inhibitory. Okay, so make it less inhibitory over time, and so then you would get more feedback to help increase the amount of power out of that. So yeah, you guys are getting way too deep. No, visualization doesn't help with cerebellar part of the learning, but it does help with the motor planning, that supplementary and premotor area. That's where it helps, is developing that plan. You can tap into that plan and sort of refine it, and you actually see activity. When you're doing visualization of an action, and we actually do this with patients who say, if you have a stroke patient who actually can't physically walk, you can have them practice walking before they're ready to walk in their head. But what they find is that you actually have to have, in order for visualization to work, you have to have done the task before and have a little bit of that program stored up there to tap into. You can't just think of a novel task and visualize it and learn it. You have to at least explore and use the task enough so you've got a little program up there to tap into, but then you can refine it through visualization and mind practice, mental practice, essentially. So we're going to talk about lobes and divisions, okay? So there's different divisions and different ways of slicing and dicing up the cerebellum. And try not to get it, it's simple, but it can be confusing, okay? So let's talk about lobes first. So there's 3 lobes. There's an anterior lobe, a posterior lobe, and a flocculonodular lobe. Here's the anterior lobe. Then all the rest of this out here, this is all posterior lobe. Then down here is the flocculonodular lobe, okay? So you've got this little anterior lobe, a massive posterior lobe, and then also a smaller flocculonodular lobe. If you look at, if you look from this standpoint, from the front, I think I have better pictures here. I'll do it on that. Then we also have, we can actually divide the cerebellum into vertical divisions. If you take a cerebellum off, you look at the front or the back of the cerebellum, and you can divide it into these vertical divisions, okay? So the vertical divisions are the vermis is the most middle. Vermis means worm-like, and this looks like a little bit of a kind of, when you'll see it in a different picture, it looks like a little bit of like a worm. Then just outside of the vermis on each side is called the paravermis. So that's this region right here is the paravermal regions. And then most of the rest of it out here is called the cerebral or lateral hemispheres or cerebellar hemispheres, but it's the lateral hemispheres of the cerebellum. So those are the vertical divisions. Then we have what's called phylogenetic divisions, which are kind of based on, you know, evolutionary, like what is the newest versus oldest parts of the cerebellum from an evolutionary standpoint as we've developed into walking, talking, moving human beings. So there's the term archicerebellum, which is synonymous with the flocculonodular lobe. Okay, so the archicerebellum is synonymous with the flocculonodular lobe. Then we have a paleocerebellum, which is synonymous with the vermal and paravermal regions. These vertical, this whole area right here in the middle is the paleocerebellum. Then the neocerebellum, the newest, most sophisticated, that helps with distal control of our body, is the lateral hemispheres out here. Okay. So the phylogenetic divisions, you know, is a mix between looking at the lobes versus the vertical divisions is how you divide that up. Then the probably most important, but you still need to know the rest, is we call the functional divisions of the cerebellum. And it makes sense by what they're called. The vestibulocerebellum is really important for coordination of muscle activity of our head and neck associated with the vestibular input. So our muscles in our head, when we make quick pendular movements of our head, are taking in lots of information from our vestibular apparatus that go into our cerebellum, and then that information is processed and makes sure those muscles turn on, turn off in a good, smooth way, dampened way, and so forth. So that's the vestibulocerebellum. Helps to coordinate head, neck movement, and eye movement, okay? Some eye movement. Well, yeah, most of your eye movement as far as like so your eyes don't jump around, so the eye movement looks smooth, okay? Then we have the spinocerebellum. Well, I'm sorry, the vestibulocerebellum is located or synonymous with the flocculonodular lobe, okay? So if you look at that, that's basically our archicerebellum. And our flocculonodular lobe are the same as our vestibulocerebellum. These guys are the same thing. Okay, so vestibulocerebellum is also the archicerebellum. It's also the flocculonodular lobe, and it has to do with coordination associated with vestibular input for both your eyes, head, and neck. Then we have the spinocerebellum, which is this vermis and paravermal regions together. Spinocerebellum, think spine, think proximal postural gross movement. So this is coordination of your trunk muscles, okay? So trunk muscle coordination is the spinocerebellum. And it's nice because it's the middle of the cerebellum. It's that vermal and paravermal regions, which I kind of think synonymous with the middle of my body, you know? And it influences not primarily it's the reticulo and some vestibulospinal tracts, so the reticulospinal tract, some of those medial activating pathways. So the way it influences coordination is the output from the cerebellar nuclei go to these medial activating pathways, okay, to influence motor output of postural and gross muscle movements. Then we have the cerebrocerebellum. Cerebrocerebellum is the lateral hemispheres. It has to do more with coordination of your distal limbs, okay, distal limb movements. So, and that's what we've been talking about so far when we've talked about this going up to the thalamus and motor cortex. The cerebrocerebellum is the one that mostly goes back up to the motor cortex because that's where you have more of your voluntary distal limb control. So the cerebellum exerts its influence over your motor system by communicating with your cortex and influencing the lateral activating pathways. Some rubrospinal, which is in your brainstem, but mostly your lateral cortical spinal tract. That's how it exerts its influence of helping to coordinate us through the lateral cortical spinal tract. So here's a nice picture here that shows, so on the picture on the right is a color-coded. So this is looking at the top. So we're looking right at the top of the cerebellum here. The pink, or not, whatever it is, red, pinkish color there is the anterior lobe. All of the green is the posterior lobe. So you can see how big that posterior lobe. This here is looking at the front of the cerebellum. It's been cut off from the pons, and we're looking at the front. We see this is the cerebellar peduncles here, where it's been cut off from the pons. They don't do a very good job of showing that superior, middle, and inferior cerebellar peduncles. But just this little blue strip right here, oh, sorry, it comes down like that. That's the flocculus, or the floccular nodular lobe. So that area is called the flocculus, the floccular nodular lobe, the archicerebellum, or the vestibulocerebellum. So that could be all four names for that one little area, the blue area there. Yeah. Vestibulocerebellum is the most important to know. Vestibulocerebellum, the functional role of that. So here is this is the anterior. This is looking again at that cut off portion of the cerebellum where it's been cut off the pons. That's the anterior lobe. Then all the rest of this down here is posterior lobe here. And then this is your floccular nodular lobe is right here. That little region in there. Okay. If I were going to show this on that lab kind of exam, visual exam, I would just outline one of those areas for you to identify. Okay. So, talk about nuclei of the cerebellum, the deep nuclei. So what happens is, information comes into your cerebellum, it's processed in the cortex. Purkinje cells are the output nuclei, the output cells. They inhibit, or they either provide more inhibition or less inhibition of the deep cerebellar nuclei. The deep cerebellar nuclei is how ultimately information gets out of the cerebellum to then go either to the thalamus and back up to the motor cortex, or to some brainstem nuclei, vestibular nuclei, to influence the descending lateral activating and medial activating systems to help adjust motor output, okay, on the fly. So starting from medial to lateral, so the most medial nuclei to the most lateral, you start with the fastigial nucleus. The fastigial nucleus projects to the vestibular and reticular nuclei, which influence the vestibulospinal and reticulospinal tracts. Those are basically all at your medial activation system, okay? Especially for the head, neck, okay? We'll show you here in a second how that's all connected. Then we have what's called the globus and embelliform nuclei, which are considered together kind of as a group because they go to the same, they influence the same areas or motor parts of our system. They are known also as the interposed nuclei because they're interposed between the fastigial and the dentate. They're between, so they're interposed. But the globus and embelliform. These actually these project to the red nucleus and the motor cortex. They actually do project to some of the lateral activating pathways. And then the dentate. The dentate projects to the red nucleus as well, but also premotor cortex and motor cortex. I should say premotor and motor cortex, but it actually does somewhat influence some of the premotor areas and even some of the motor planning areas to help you in that motor learning process, right? The premotor areas is where you store some of these programs. That's helping you store the right program up there so that the next time you do it, it's closer to what you hoped it to be. So now we're looking down at the cerebellum, and actually the pons would be, well, actually I take that back. This is looking at the front of the cerebellum, and this is a frontal view or a coronal view through the cerebellum, okay? This is the top, bottom, or dorsum, or superior and inferior surfaces of that, and then we're looking straight at, and so we're cut through, looking at kind of a mid-coronal frontal section. Here's the dent. So let's go medial to lateral. So fastigial is this nuclei right here on each side. You have pairs of these. So fastigial nucleus, and then you have the globus and embelliform nuclei here, the interposed nuclei, and then here's this dentate that goes, and it's pretty big and wraps all the way and sticks farthest out into the lateral cerebral hemispheres. But you can see it also crosses somewhat into the vermal and paravermal regions. So, but mostly associated with lateral hemisphere function. This is actually showing that in a cross-section where somebody really took some time to carve out the white matter around some of the nuclei. And this is actually the globus and embelliform nuclei right there, the interposed nuclei. This is your dentate that is all around here. And then this is your fastigial nucleus here and vestigial nucleus here on this side. Let's take a break right there. Just that kind of covers some of the basic functions of anatomy. Then we'll start drawing how that goes through there and hopefully make it less hard. And so we got to get information into the cerebellum, and that is through the afferents, the mossy fibers, climbing fibers, okay? Those are going to bring information in through the cerebellar peduncles, okay? Via the peduncles. Those afferents go in and they synapse in the cortex, okay? Those mossy and climbing fibers go up to the cortex to influence Purkinje cells, okay? Hey guys. The synapse in the cortex. Purkinje cells relay info from the cortex then to the deep cerebellar nuclei, those vestigial, embelliform, and so forth. And then the deep cerebellar nuclei is where the efferents that leave now the cerebellum to go influence the cortex, influence the brainstem, okay, and the descending motor pathways. All right. Let's see here. Okay. So this is showing you information how it gets the afferents and the afferents and the efferents, just in a more simplistic fashion here. So here is, and this is just most, this is not every detail, but just mostly, and this is as much as you need to know. So most of the information coming in from your proprioceptors, your vestibular system, most of it comes in via the inferior cerebellar peduncle. You also have some information that comes in through the middle cerebellar peduncle. This is the copy of some of the motor messages, the voluntary motor messages coming from the cerebral cortex. Those are called the cortical pontine fibers, which are running right along with your cortical spinal tract fibers. So your cortical spinal tract fibers that come down through the front of the pons that are going to end up becoming your lateral and medial cortical spinal tracts, they have collaterals that branch off and interact with these called pontine nuclei in the pons that then send an afferent signal or a copy of those motor signals through the middle cerebellar peduncle to the cortex. And these are going to go to the cortex. Okay? Then the way you influence or get out of the cerebellum is once you've processed information in the cerebellar cortex, that signal, Purkinje cells interact with the dentate, interposed, and vestigial nuclei. They adjust the level of activity of those, and those output, most of the output goes out of the superior cerebellar peduncle and then goes to thalamus, red nucleus, vestibular nuclei, also reticular nuclei to influence our motor pathways, both our lateral activating pathways and our medial activating pathways. That's how it gets out. Now, if you'll recall, we talked about a spinocerebellar pathway that some of that information went in through the superior cerebellar peduncle. So there's some exceptions, but this is more the general rule. You don't need to know the exceptions. Okay, know the general rules more than the exceptions. So let me just draw this out a little bit for you to kind of, and we're going to use the example of more of the lateral hemispheres of the cerebellum, which have to do more with your distal voluntary control. Let's start with that, okay? So the lateral distal coordination, voluntary movement coordination, okay? Let's start with you sending a signal to your muscle to do some activity, okay? So it starts up, let's start up here in our cortex. So remember this, first of all, you think about doing the activity, has to go through the basal ganglia first. Then it goes up to the primary motor cortex to release the signal to start going down to the muscle, okay? But I'm going to erase the basal ganglia loop. Just remember that's in there. So we're sending a signal from our primary motor cortex. This would be more, that'd be more trunk up here. But let's do a hand. let's move out here to the hand region. So maybe right in here. Comes down through the corona radiata, internal capsule, crus cerebri, which is the front of the midbrain. So this is midbrain here, okay? And this area is pons right here. And then medulla below. We have our two cerebellar hemispheres sticking out there, okay? I'm going to get rid of some of those lines just to make it less confusing. Why won't that go away? That's weird. Never seen that before. Oh. Hide. Okay. That's okay, got it. All right, so this signal's coming down. There are, in the front of the pons, there's these little pontine nuclei, okay? So we're just going to have one pontine nuclei to show you here. Some of these keep going down, but some of this interacts with this pontine nuclei. I'm going to change colors here. Let's go to blue. So let's change that pontine nuclei to blue there. A copy of whatever motor signal gets sent to that pontine nuclei. That pontine nuclei sends its signal. In this case, it's via the middle cerebellar peduncle over to the cortex of the cerebellum. So you got to copy that message going in there to tell that cerebellum a little bit about what's happening with the signal you're planning on sending down to that location. We also have, I'm going to do this maybe in different color. Let's do green here. Down in the medulla, we have the inferior olivary nucleus down here, right? It also receives a copy of the motor signal that you're going to be sending to it. And then this is going to go down and it's going to cross over in your lower medulla, and we're going to say that's your lateral cortical spinal tract. It's going to go deliver a signal to your arm to make you, it's going to go down in your spinal cord into a lower motor neuron, which is going to go out to your hand or arm out here, okay? So your arm is moving, and your muscles are being stretched or shortened, and your joint receptors are being activated, and it's telling you, that information is telling your body about what's happening with the result of that movement. And so let's do another, let's do orange or something like that. So that information comes in via, we're just going to primarily right now say spinocerebellar tracts as a group. Just group them together right here, spinocerebellar tracts. The spinocerebellar tracts come up here. They actually deliver a copy over here to the inferior olivary nucleus. Not that important to know that, but just want you to see that. But mostly what's important is they come in through the inferior cerebellar peduncle. This is via the, these are mossy fibers, right? These are also generally considered mossy fibers, these up here too, the cortical pontine. So all those are coming in are the mossy fibers. We also have, let's do back to green here. I think I used the lighter green last night, lighter green. This is climbing fibers coming in here from the inferior olivary nucleus. So you'll notice that your inferior olivary nucleus gets a copy of the motor signal, but it's also getting all the proprioceptive information to it as well. The inferior olivary nucleus's role is over the long term to actually set more the long-term sensitivity of the Purkinje cell, how inhibitory it is. Is it more inhibitory or less inhibitory at any given moment, but at a long-term basis? And that's based on kind of repetition as you're moving your arm and so forth. Then those climbing fibers help say, you know what, I use my arm this way, you know, for this activity, and so I need my Purkinje cell to be set at this level of excitation for this particular activity because I've learned from experience that it needs to be ramped up or ramped down, okay? So I'm going to erase that one, just get it out of the way right now. But you also get this signal here that comes from the mossy fibers that influence the more short-term. The inferior olivary nucleus has to do with more of the long-term regulation of the sensitivity to the Purkinje cell, whereas these mossy fibers coming in have to do more with the moment-by-moment changes that are occurring, okay? So what happens is your motor signal that came in, you get a copy of the motor signal from the pontine fibers, from the pontine nuclei, sends a signal. That information, then a signal comes from the mossy fibers. There's some comparison going on there between those signals, just like the comparison was made in here. Is there a match? And this is also saying, is there a match between these signals based on how excitatory or inhibitory influences are being all converging within that cerebellar cortex? If there's usually some slight mismatch, unless you're really good and really learned it. But let's say this is a new thing you're learning, so there's a mismatch. And your sense is that the signals that you're getting from your motor system and the feedback you're getting from your proprioceptive system are saying that what you actually did is not quite the same as what you intended to do. Because this initial signal, this is your intention. Because what's going to happen is that signal is what you intended to do. That it's sensing what will ultimately end up at your lower motor neuron. But then as you move, that what you thought you were going to do might slightly be different, because you sent a signal that said move my arm this amount or this with this force, and now it's moving quicker than you expected, or it's moving more slowly than you expected, or there's more tension developing in the muscle or less tension you developed in the muscle. So I need to adjust the next signal, okay? So that comparison happens in the cerebellar hemispheres, but also in the inferior olivary nucleus. All that information converges, though, in the cerebellar hemisphere. Then you go, okay, I need to adjust the next signal coming down. So what you do is you send a signal from the Purkinje cell. Let me just do a different color here. Let's do red. No, let's do purple. So here's the Purkinje cell, big Purkinje cell. It projects to, in this case, this is going to be the dentate, right? So the dentate nucleus is that squirrely thing in here. Projects to the dentate. It influences the dentate and the signal that it's going to send back to the thalamus. So let's draw a thalamus in here. Remember, we have a thalamus on each side, but there's our thalamus. So we'll go back to purple here. This is going to project back. It's going to cross over in the pons, go up, communicate with the thalamus. Then the thalamus is going to go back to the cortex, and so that the next signal you send down will be adjusted up or down from there. But you see here, so that, remember we talked about all these spinal cerebellar tracts are ipsilateral, but they still influence the opposite cortex because of this crossing over here, okay? But if you have damage to a cerebellum on this side, it still influences the same. Because you've damaged these ipsilateral pathways coming in from that limb, but it also damages and prevents this error correction happening to that side, the opposite side of the cortex, because these cortical spinal tracts cross back over. The impairments in coordination happen ipsilaterally. So you have to know that if you have left cerebellar damage, you're going to have left coordination problems. If it happens to be in the lateral hemisphere, it's going to be more distal coordination. If it happens more in the vermal, paravermal, it's going to be more truncal in coordination, but on that left side. If it happens in the flocculonodular lobe, just on the left side, your left eye and your left neck and so forth will have less coordination. So cerebellar lesions still cause ipsilateral problems. However, a cortex lesion will cause a contralateral problem. Contralateral, as it means, related to where the lesion is, okay? You have the similar setup and pathway for, that was lateral hemispheres, that's distal, fractionated distal voluntary movement. You have the similar thing for more postural and gross movement of your trunk, and the same thing for your eyes and head and neck movement. That same process for those muscles that are more for eyes, head, and neck, and then trunk muscles. So that's what we just showed you, was the connections for the lateral hemispheres. Okay, so for the this is the cerebrocerebellar circuitry I just drew out. It's going back up to the cerebrum to influence the cerebrum. There's also like the vestibulocerebellar nuclei or system that has a similar type of influence. but influences. So here's the, let's just draw that out really briefly here. Let's go, let's make this the vestibular, here's your vestibular nuclei right here. Those are in your pontomedullary junction right here, and you have vestibular nuclei on both sides. Vestibular nuclei are so intimately related to the cerebellum, they're almost considered another cerebellar nuclei. They almost function like a deep cerebellar nucleus. And you'll see here they actually do in a sense. So here's your vestibular apparatus. Yeah. Actually, your vestibular apparatus is 3 semicircular canals, kind of something like this. But so this is a vestibular system. Signals come into the vestibular nuclei. Then the vestibular nuclei send that signal to your cerebellar hemisphere. Okay? That information is processed. This is non-conscious information, so there's really not like conscious signals coming down in there. Although if you're doing voluntary head movement, you would get some signals coming down from your cortex, from your head. Come down. It could impact some pontine nuclei that would send that signal here if you're doing more voluntary head movement. Okay. Those signals are compared. It could then send a signal both back up to the cortex to influence voluntary head movement, but most of the vestibular stuff is non-voluntary. So it's really more of just, it's a very simple, a little more simplified loop in that it's just goes from the vestibular nuclei to the vestibular cortex, or to the I shouldn't draw that way out in the periphery there. This is vestibular. That would go to the floccular nodular lobe. So we have to kind of like make this assumption that we're looking at floccular nodular lobe. So that's the cortex and the floccular nodular lobe. Vestigial. I'm drawing that a little too far out, so I should draw this relative to vestigial nucleus is what that vestibulocerebellum would be influencing. Then from the vestigial, it would go back and influence the vestibular nuclei again to influence the vestibulospinal tracts that start in the vestibular nuclei. And remember, you have the vestibular nuclei. We haven't gone into this in detail yet, but it's got 4 different subdivisions of nuclei. Part of the reason you have those 4 different subdivisions is because there's lots of activity happening in that vestibular nucleus. It's acting as a primary initial point of reception where information comes in. It's relaying that to the cerebellum. Cerebellum is relaying that information back to the vestibular nuclei. Then the vestibular nuclei are sending signals down to proximal head, upper back, neck muscles to influence the coordination of those as it relates to your head movement. All right. So let's go through a little bit of that again here. Functional divisions of the cerebellum. These are the most important to know. The vestibulocerebellum, which I just started drawing out there. It's also known as the floccular nodular lobe or the archicerebellum, are all synonymous there. Sometimes it's also just known as the flocculus. It plays a role in coordination of eye and some postural movement, especially of the head and neck. Okay, so eye movement, head and neck movements. It helps those muscles work appropriately in a coordinated fashion. It projects to both vestigial and vestibular nuclei. Okay, so I didn't show you here. It also projects to the vestibular nuclei and the vestigial nucleus, which I guess I did show that there. The way it exerts its influence over coordination of those is via the vestibulospinal tracts. That's how it's output. That's how it gets to adjust the activity level in your neck and upper back area related to the information coming in from the vestibular system. So it makes sense. Vestibulocerebellum, it exerts its influence through vestibulospinal tracts. Then we have the spinal cerebellum, which we mentioned is the vermal and paravermal regions, also known as the paleocerebellum. The vermal region, the very center part of your cerebellum, has to do more with influencing coordination of trunk, proximal limbs, and speech, your muscles of your mouth. All your muscles in your body have ties to the cerebellar system, okay? Most of our muscles move quickly and need some dampening and motor learning to occur, okay? All of our muscles, really. The paravermal region, so the spinal cerebellum includes the vermis and the paravermis. The paravermal region has to do more with the real proximal limb musculature. The vermis, it's a little more distal. It's still kind of proximal limb muscle, but this is real proximal limb muscles like shoulder blade, girdle muscles, your butt muscles, and those kind of things, and your spine and those kind of things, your multifidi and erector spinae and those kind of things is more that paravermal region. It just kind of makes sense. It just starts more laterally and you get more central with that. So it starts out, but it's the cerebrocerebellum that's more the real distal stuff, okay? The way the spinal cerebellum, those areas project to the fastigial and the interposed nuclei, but mostly the interposed nuclei. But there's some that go to the fastigial, but it's mostly interposed nuclei. The spinal cerebellum is interesting because it's kind of in the middle, so it actually there's a lot of overlap with what it does, okay, in your body. So it actually influences almost all the pathways in some way to control movement of the body. It influences the vestibulospinal tracts for the proximal and trunk muscles of the upper back and so forth. The reticulospinal tracts for, again, proximal muscles all through the trunk and into the lower body, and the rubrospinal tracts for the upper body, as well as the medial and lateral cortical spinal tracts. It has some influence over those. It tends to influence more the medial activating pathways and a little less of the other lateral activating pathways. So it has a heavier influence over vestibulo, reticulo, and medial cortical spinal tract, and probably a little less over the rubro and lateral, but it has some. It's kind of in this transitionary area of your coordination with your cerebellum. And then more lateral is the cerebral hemisphere, or cerebrocerebellum, and it communicates the cerebral hemispheres, or I'm sorry, it does, but it's also called the cerebellar hemispheres or the neocerebellum, or some names you can also call it cerebrocerebellum. It's most important for coordination of distal limb movement, okay? Also influences some of the motor planning and programming, okay? Especially for voluntary motor activity, purposeful voluntary motor activity. It sends the signals, or the area of the cortex and the cerebrocerebellum, those Purkinje cells, project to the dentate nucleus. That's that pathway I just drew out for you guys. This is nice as it kind of shows you the functional relationships here in a color-coded fashion here. So you see the vestibular apparatus is very closely associated with the vestibulocerebellum. The spinocerebellum is very closely associated with the spinal cord and the influencing spinal pathways. And then the lateral hemispheres are communicating with the cerebral hemispheres up in the cerebrum. They're making connections with those. So just kind of if you're a visual person, and just kind of, that helps kind of tie them together, which is influencing which the most. And it's not that simple, like anything. There's gray areas, there's overlap in these, but there's kind of like a homunculus of your cerebellum. So this is kind of the homunculus of your cerebellum. So you can see kind of your spine, and your proximal area and spine and girdle muscles in this vermal, paravermal regions. And I don't know why they don't show those go farther out, but here's your distal muscular projecting into the lateral hemispheres. Here's your eyes, but your floccular nodular lobe is in here. And that's where your eyes and coordination, your eyes, the vestibular system is located. And then your speech, this is supposed to be speech. Your mouth is right in that vermal region, okay? So you can kind of, that picture again kind of helps you just picture where the body's laying across. You can just kind of picture a body laying through the middle of the cerebellum, kind of like this. And just know that the eyes, though, are kind of in the middle associated with that floccular nodular lobe. So what this is now, this is from your book, and this is just showing you the vestibular output. How it gets from the nuclei, the deep cerebellar nuclei, to actually influence motor pathways, okay? So we're ignoring how it got into there, cortex, how it's processed in the cortex. This is just now the Purkinje cell has sent its signal to a deep cerebellar nuclei, and that deep cerebellar nuclei is going to communicate or influence descending motor pathways. So this is showing you, this is vestibulocerebellar output. This is vestigial nucleus, although vestigial nucleus here, and it's projecting to vestibular nuclei, and it actually projects somewhat bilaterally. Don't need to know that, but just that it projects to the vestibular nuclei to influence the lateral and medial vestibulospinal tracts to help with the neck control associated with the vestibular system, providing proprioceptive input about your head position so that when you make adjustments to your head, it's nice and smooth and doesn't look uncoordinated. This is spinocerebellar. And remember when I showed you the spinocerebellar influences lots of different areas because it's kind of in the middle and it functions and plays a lot of roles here. So this is how it exerts some of its influence. So actually this is the Purkinje cell. This is the cortex Purkinje cell projecting to the vestigial nucleus, but it also influences the globus and umbelliform nucleus. Okay, so they should put globus and umbelliform nuclei in here as well. So the spinocerebellar output goes to all those nuclei, and then it can influence the vestibulospinal tracts, the medial and lateral vestibulospinal tracts, because it has connections from those that project to, the vestigial nucleus projects to the vestibular nuclei. It influences the reticulospinal tract, because it actually has projections to the reticulospinal tract. Which you get, just remember reticulospinal tracts are mainly for motor activity of the trunk and proximal muscles. However, it does influence a little bit of primarily the medial cortical spinal tract. So some of those do project actually up to the thalamus to influence the medial cortical spinal tract, which is again another medial activating system. So again, more posture, gross movement. But it also projects a little bit to the lateral cortical spinal tract as well, okay? Don't worry about that aspect so much. Think spinocerebellum, posture, gross movement, proximal stuff, coordination of those muscles. Vestibulocerebellum, head and neck, and eyes associated with the vestibular system. And then cerebrocerebellum, distal movement, coordination, okay? So here's the Purkinje cell projecting to the dentate. Then it projects up to, it can influence the red nucleus and the rubrospinal tract, which is that distal flexion of the upper extremity. But it also projects to the thalamus, then projects to the cortex, which then can influence the lateral cortical spinal tract. The signals coming down the lateral cortical spinal tract. So that's the output of the cerebrocerebellum. So what happens when you have damage? So lesions of the cerebellum. If you have it in the vestibulocerebellum, so that floccular nodular lobe, vestibulocerebellum, if there's select damage in those connections or in that cerebral portion of the cerebellum, you will often, the couple key symptoms will be some type of nystagmus, where as you move your eyes, they will jump, okay? And you'll see this jumping of the eyes or beating of the eyes as they move, because you can't turn the muscle on and turn it off in a smooth, dampened way. So your muscle will turn on, but won't turn off quick enough, and your eyes slide past where you want them to go, and it looks like it's jumping or beating. That's called nystagmus. The other thing the person may have trouble with is just equilibrium. They feel out of balance, especially because they can't match up head and neck position and coordination of the head and neck related to the vestibular input. So they feel a little swimmy. They feel like, ooh, I just feel like I can't quite, because my head and neck muscles aren't syncing up with my vestibular system well enough and coordinated enough to make me feel stable, okay? If you have spinal cerebellar, so this is the vermal and paravermal regions, if you have specifically a lesion that involves just that very central part, that vermal region, they will often have dysarthria, like they'll have ataxia and incoordination of muscles of the mouth to coordinate speech. Remember that little speech area, the mouth is right in that center. So it'll sound dysarthric, but I can't imitate it, but there's a type of cerebellar dysarthria that it's like you know it when you see it, or hear it, I should say. But they also have a lot of trunk ataxia. As long as they're sitting in a chair or something where their trunk is stabilized, they look fine. But as soon as they sit forward, they can't, and their error detection system is off, and they're over-correcting or under-correcting with their trunk muscles because they're just, the information from the proprioceptors and so forth is not going back and processing in the cerebellum very well. If they have more paravermal, that tends to be more of these kind of muscles, proximal muscles in here, they'll have a lot of gait ataxia. They can't control the proximal musculature, and they also have some equilibrium problems, not because their head feels swimmy, but just because they can't maintain really good postural control, especially during gait. However, usually you have both of these. It's hard to just lesion the vermis and not the paravermis. Usually you get a mix, okay? But people if it's pure spinocerebellar problems, they might do decently with like this kind of stuff. If it's just moving the distal limb, they might do fine with finger to nose or rapid alternating movement. But once you start making their proximal system have to work and stabilize, now they may have trouble. Like if you sit them on the edge of a table and their trunk is doing this, it'll throw off their arm. But if you put them on a chair or a wheelchair where their back is stabilized, then they look like they have pretty good distal coordination. Do you have a question? That can be one way to compensate an abdominal binder. You give them and that abdominal binder provides actual physical restriction of movement, but it also provides some extra proprioceptive feedback that makes conscious level awareness that they might be able to, correct consciously some of that as well. So, yeah. It's the same principle, like if somebody's got a lot of truncal ataxia and they're having trouble in standing position, putting braces on their feet to lock their ankles in place and just at least take the ankles. That's one less joint they have to control, and it dampens their ataxia because you've got a physical solid thing, bracing them. And then cerebrocerebellum, that's where you get the distal. Like, they can't do the rapid alternating movement. They're having trouble getting to their nose and that kind of stuff. Okay, or the heel to shin is really a challenge for them. That's more the cerebrocerebellum. Most lesions are asymmetrical and get a couple different regions, so you usually get a mix. But just know that if it happens on one side of the cerebellum and doesn't cross the center, the midline of the cerebellum, the deficits are all going to be on that same side as the lesion. Okay, so right side lesion, right-sided problems, left-sided lesion, left-sided problems. So the impairments, the key term here is ataxia. It's a general term, overarching term, just meaning lack of coordination, if you use the term ataxia. Then we can talk about a little more specific descriptors or subcategories of ataxia. One is called dysmetria. Dysmetria is the inability to accurately move an intended distance. And it's usually a combination of both hyper and hypometria. So you ask them to touch their finger, so hold your finger up. Hypo, I'm not quite getting there. That's hyper, I go past it. So it's usually a combination. They can't make the error correction to really get in there and adjust and dampen the muscles to make it look smooth and turn the muscle off in a smooth way right when it gets to that point. So although a lot of people can use vision to help do that and dampen some of that, it's when they, and then if you have their eyes closed and they bring it to their nose, that's where you can sometimes really notice it more with their eyes closed, because they can't use vision and conscious, guidance, and they really start to, poking themselves in the face, and they can't. That's hyper would be they're poking themselves in the face. Hypo, they're not quite getting there. So that's dysmetria. A term that is called dysdiodocokinesia. That's a mouthful. Dysdiodocokinesia. Kinesia, moving your limbs at the same time in a rapid way. This is dysfunction. So dysfunction of co-movement of limbs. So they can't do this. They can't do the rapid alternating. You can say patient has impairment in rapid alternating movement, or heel to shin, or you can say, if you want to, if you know the spelling, patient demonstrates dysdiodocokinesia, moderate dysdiodocokinesia in the upper extremities. Then the other thing, yeah. They still may know, because they still have conscious proprioception, most of them, and so they're trying to get there. They know they're not there, but just their cerebellar system isn't working enough to help them get it there the first time. Then they finally try to get there, but then they go past. Now it's too far, and that's hypermetria. So action tremor. So remember Parkinson's patients? They have a resting tremor. They have this resting tremor. People with cerebellar problems, as long as they're like stable and the hands are on their lap or whatever, they don't have a tremor. But when you start moving, and the closer they get to the target and have to really get it, fine-tune it, then it gets worse. That's called an action tremor. Most Parkinson's patients, when they move, it goes away because they don't have a cerebellar problem. It's a basal ganglia issue that's causing that tremor. When they start moving, the tremor goes away. Cerebellum, they look good at rest, but then they start moving and they have an action tremor. Yeah.
You compare that, say, if you just got done working out and you've had a really hard workout and you're kind of like shaking.
Yeah.
Is there any correlation to like where that cerebellar or anything?
That's usually just more motor fatigue.
Motor fatigue.
Yeah, so it's just more of a fatigue issue. It doesn't really have to do with cerebellar issue or some temporary loss of cerebellar proprioceptive. It's just more of a fatigue issue at that point. Yeah, if you really depleted your muscle and it just has trouble firing effectively, even if you're sending the right signals to it. So good question though. Yeah, don't work out that hard, man. You shouldn't be working. Hypotonia. So people with cerebellar problems often have just a little bit of lower tone and a little bit more weakness, because remember I talked about that deep excitatory loop. So when you initially fire the muscle, that signal goes into the cerebellum, and there's that initial deep excitatory loop. It kind of boosts the signal a little bit. And so they don't have that booster signal. So they might have a little, they still have voluntary movement, but it just doesn't get boosted the right way. So it appears to be a little weak, and they might be a little hypotonic. Their reflexes aren't quite as strong, but it's not usually totally abnormal. You have to kind of like look for it. I think this is the last slide, maybe. This is from, is this in your, this is from the 4th edition, I think. I don't think does this graphic look familiar from the 3rd edition? I don't think this is in the 3rd edition. I think it's in the 4th edition. This shows you the entire motor system that includes the basal ganglia, the cerebellum, the descending motor pathways, how they all interact. Don't have to know all the connections. It's just good to sometimes appreciate the big picture. So starting on the top here, this is your cortex, which is where your premotor, supplementary, and primary motor areas that influence descending pathways. This is just showing right here, this is the basal ganglia loop. Let me show you that. This is the basal ganglia loop right here. That's all that's representing. So the basal ganglia loop, remember, it goes back up to the cortex, but it also influences the PPN is the pedunculopontine nucleus. There's another nucleus that I think your third edition doesn't mention. It's the locomotor nucleus in the reticular formation. There's another nucleus that tends to drive the automatic nature of walking, helps to drive that. The basal ganglia influences that locomotor region as well, in addition to the pedunculopontine nucleus. That's why people with Parkinson's disease, because of the influence over the pedunculopontine nucleus, they have the rigidity, but they also have this fascinating gait where they struggle to walk and they do this. That's because of the influence over this locomotor region in the medulla, the medullary locomotor region in the reticular formation of the medulla is because of that. Then the voluntary, the bradykinesia, akinesia is because of the influence or problems up to the motor cortex and the voluntary influence there. These are both in the reticular formation, and they both exert their influence through the reticulospinal tracts. Those medial activating pathways. Here's our lateral cortical spinal tract that goes actually down to the lateral motor neuron pool for the distal movement. This is the rubrospinal tract that goes through the red nucleus down to the lateral motor neuron pool, especially flexors of upper extremities. Here's our cerebellum. Cerebellum's receiving information from the vestibular nuclei. The thing they don't show here in the cerebellum, it's perceiving all that body information from muscle spindles, joint receptors, all that stuff. The cerebellum influences the motor cortex. It also influences both ways the vestibular nuclei via the vestibulospinal tracts. Cerebellum also influences back up to the cortex for coordination. Also influences the reticulospinal tracts. And then this is the medial motor neuron pool. This is the medial activating system. And these are some of the things it does. Adjusting muscle tone, stepping movements, contraction of neck muscles and extensor muscles, and shoulder and trunk muscles, and those kind of things. That's kind of the big picture of all of what you just learned. Just imagine, so if it makes you feel any better, at least you're only having to take a test just on the motor system, because previously we did both somatosensory, the sensory pathways, plus all the motor system on one exam. So that's the reason why we did change that to that. So, all right, let's talk about. I think I'm going to pause this here. I'm going to finish this